Should we start? Yeah, okay. So welcome everyone, good morning. Today it's my pleasure to introduce Anthony for giving his final dissertation defense. Anthony joined my lab when we were still in Maine at MDI Biological Laboratory. And that was in 2017, after he was accepted into the University of Maine graduate program. This program is exceptional in a way because the students there don't rotate within just a single department, but they're actually rotating in the entire state of Maine. And this is because Maine is such a huge state with very few research institutions that there's really just one graduate program for the whole state, which is quite fascinating. And so what typically happens is that the students stay either in the northern part or in the southern part. But Anthony stayed in the northern part, which is where the Jackson Laboratory is, and MDI Biological Laboratory. And I was glad and happy that Anthony decided to come to my lab. And so shortly after that, unfortunately, I decided I'm going to move to Miami. And so Anthony was the only person in the lab who wanted to come with me. He was actually very happy about that because he came originally from Chicago and he really liked the warm weather. so he, unfortunately, I think his whole PhD was a sequence of unfortunate events. Because of the move, he lost actually probably around 8 months of his time because we didn't get the equipment right away. So he had to wait a long time until we got settled in here and were ready to do our research. And then he also had to repeat quite a lot of classes, even though he was almost finished with his credits in the University of Maine program. He had to repeat some of those here, so he really didn't get started for quite a while. And finally, when he thought he was finished, with all these hurdles, COVID struck. So then he again lost probably 6 months with social distancing and so on. So he was probably the most unfortunate student in this program ever. But despite all these problems, he actually got to, he managed to get a fellowship to go to Woods Hole to participate in one of their prestigious summer courses on zebrafish genetics. And he also was able to obtain a scholarship from Japan to go to a Japanese lab, or Kawakami lab, where he was able to screen transgenic zebrafish for identifying some of those fish that were useful for his own research project. And among other various awards that he received during his PhD, he was awarded one of the most prestigious awards from the Peripheral Nerve Society, the J.W. Griffin Award, which was given to him for his research that he presented at the annual meeting in Italy in 2019. And all, despite these hurdles, Anthony published a total of 3 first author publications, and he will have another co-authorship in a 4th paper that is currently being written and should be ready for submission in a few weeks. So Anthony's PhD project is focused on understanding how paclitaxel, which is a chemotherapeutic agent, promotes sensory axon degeneration in the skin. And he was using primarily zebrafish for this research. So I won't tell you too much about it and rather have Anthony give or present the story to us. So I would like to welcome Anthony and hand over to you.
Thank you so much. That was such a nice introduction. Okay, so it's my pleasure today to present my work. The title is The Role of Epidermal Hydrogen Peroxide in Paclitaxel-Induced Cutaneous Sensory Axon Degeneration. Thank you all for being here. As Sandra stated, it's been quite a journey. So it's really nice to finally be here presenting all this work. So the overview of my presentation, I'm going to go through the chapters of my dissertation. And to start, we're going to go through chapter 1, which is an introduction to chemotherapy-induced peripheral neuropathy. So cancer is one of the leading causes of death worldwide. If you look here at the statistics from the American Cancer Society, it is estimated in the year 2022, there will be an estimated approximately 1.9 to 2 million new cases of cancer. And there will be approximately about 600,000 estimated deaths due to cancer. However, despite these high numbers, due to advancements in technology In medicine, we're able to detect cancer much earlier on, and we were able to treat it way more effectively. Now, a majority of cancer patients, due to the treatment that they receive, experience toxic side effects. And one of the leading toxic side effects that occurs in a majority of cancer patients is peripheral neuropathy. And peripheral neuropathy is a disease of the peripheral nervous system. So we have a central nervous system, which consists of your brain and your spinal cord. And then you have your peripheral nervous system, which consists of your peripheral nerves that extend out towards various extremities of the body, including your hands and feet. And you can see here how the peripheral nerves kind of extend and branch throughout the entire body. Let me get my laser pointer here. Now, chemotherapy-induced peripheral neuropathy occurs in a majority of cancer patients among many different anti-cancer agents. And despite these anti-cancer agents differing in their mechanisms of action, it is common that the peripheral neuropathy symptoms among cancer patients experiencing chemotherapy-induced peripheral neuropathy are pain, tingling, and numbness in the hands and feet. And this is like a burning sensation, it's very intense sensation in the hands and feet. And so what I'm showing here is a diagram of the spinal cord and a nerve bundle. And you can, what I'm showing here is that I'm showing how different chemotherapeutic agents that are used as common first-line treatments are hypothesized to target different parts of the sensory neurons or motor neurons or some parts of these nerve bundles. And it is hypothesized that these different targets, such as organelles like mitochondria or cytoskeletal elements like microtubules, it's hypothesized that targeting these different elements are what cause nerve dysfunction, although the clear mechanism is not really known. So one of the common first-line treatments to treat cancer is paclitaxel. Paclitaxel is a natural product. It's isolated from the Pacific yew tree. And the way paclitaxel works is that paclitaxel binds to the cytoskeletal elements found in all eukaryotic cells, known as microtubules. Microtubules are constantly polymerizing and depolymerizing. And so what paclitaxel does is it binds to and stabilizes microtubules. Now, paclitaxel is a first-line treatment for breast, ovarian, lung, pancreatic, and prostate cancer. So it is used in a wide variety of cancers as a first-line treatment, making it a really good drug to study. And so, like I said, paclitaxel binds to and stabilizes microtubules. And this is very important because microtubules are essential for many functions in the cell for the cell survival. And one very important function is cell division. And during cell division, microtubules are attaching to chromosomes and ensuring that they are properly separated into two daughter cells to ensure proper cell division. And so by binding to and stabilizing microtubules, paclitaxel inhibits cancer cell division, and this is how paclitaxel fights cancer. But of course, since microtubules are present in all eukaryotic cells, you can imagine there will be off-target effects. And this is how paclitaxel leads to one of the main side effects that occur in patients who receive paclitaxel, which is, like I mentioned, peripheral neuropathy. So paclitaxel-induced neuropathy occurs in an estimated 70% of patients who receive paclitaxel. And like other chemotherapy-induced peripheral neuropathies, this is a peripheral neuropathy that starts in the extremities. So it's known as a distal symmetric polyneuropathy. So it starts in the hands and feet, and then it progresses toward the midline. And as I mentioned before, this is characterized by intense pain, tingling, numbness, and cold sensitivity in the hands and feet. And patients can experience either all of these symptoms or just a certain combination of these symptoms. And these symptoms can tend to cycle in and out. So it's never consistent from patient to patient. And so it is known that, or is hypothesized that this paclitaxel-induced peripheral neuropathy occurs due to the degeneration of somatosensory nerve endings that are innervating the epidermis in the hands and feet. And so paclitaxel is causing a degeneration of these terminal nerve endings that are ensheathed by the epidermis. How exactly paclitaxel is causing peripheral nerve degeneration is not known. So there is no cure. So the treatment options are limited to dose reduction, discontinuation of treatment altogether, or just mitigating the pain with analgesic compounds. And all of these treatment options, you can probably guess, are just further lowering the patient's quality of life. So finding some sort of therapeutic intervention for this condition is very imperative. So what do we know so far from clinical studies about paclitaxel-induced peripheral neuropathy? So as I mentioned before, paclitaxel causes the degeneration of somatosensory nerve endings. And I want to emphasize also that other chemotherapeutic agents, despite their mechanism of action, are causing the similar kind of pathogenesis. And so here's some evidence of that. In what you're seeing are sections of patient skin. You can see, and if you focus on the right side of this panel first, you can see this is the kind of what's depicted in the cartoon. You have the epidermis, and you can see in green, these are the sensory nerve endings innervating the epidermis here. But you can see in a patient on the left with chemotherapy-induced peripheral neuropathy that there is a lack of terminal nerve endings, demonstrating that there is a degeneration in the extremities. So what do we know about paclitaxel-induced neuropathy from rodent models? So in the spinal cord, you have what are known as the dorsal and ventral roots. And the dorsal root is where there are nerve bundles for sensory neurons, which go on to innervate the extremities, such as the skin, to deliver touch sensation or thermal sensation. And then in the ventral root of the spinal cord, you have the motor neurons, which are going on to innervate muscle fibers to induce contraction or fine motor movements. And when looked at in rats that were treated with paclitaxel, you can see in the dorsal root in the vehicle versus Taxol that compared to the control, Taxol shows a degeneration of these nerve fibers, whereas in the ventral root, there is not much, if at all, difference in the nerve fiber morphology, showing that, as described in patients, that this is a predominantly sensory neuropathy. And so how is it suspected that paclitaxel is causing peripheral nerve damage? Well, one idea is that paclitaxel is damaging mitochondria, and mitochondria are organelles in the cell responsible for producing energy. They're known as the powerhouse of the cell, right? So it is thought that maybe paclitaxel interferes with energy production, and that is how sensory nerves go on to die. So here in a rodent, you are seeing also, as I showed in the human, that paclitaxel causes degeneration of terminal nerve fibers. So here's a cross section of the epidermis. In dark brown, here are the sensory nerves innervating the epidermis, and then you see in the paclitaxel treated animal there are little to no nerve terminals above this dotted line. And when quantified, it's shown that there's a significant decrease in the nerve fibers in the paclitaxel treated animals. And so when looked closely in the sensory nerves or sensory axons, looking at the mitochondria morphology, this is using transmission electron microscopy, looking at the tibial nerve. These mitochondria you can see are pretty electron dense compared to the paclitaxel treated animals. So if we look at the tibial nerve in the paclitaxel treated animal, you also see evidence of these white spots, which are known as vacuoles, and vacuoles are indicative of mitochondrial damage. And so when also looking at the mitochondrial diameter, we are looking here at unmyelinated axons and then myelinated axons, and also Schwann cells as a control. And we can see in the unmyelinated and myelinated axons that paclitaxel treated animals have mitochondrial diameters which are significantly increased, and swollen and vacuolated mitochondria are a sign of damaged mitochondria. And this is very interesting because paclitaxel has been also shown to cause similar effects in mitochondria in vitro or in isolated mitochondria. So it's interesting to see the effects of Taxol happen in isolated mitochondria and also in vivo in the rodent animal. Further studies into the dysfunction that might be caused to mitochondria by paclitaxel have been done. And so if we look at a sensory neuron, we can see it's a very polarized structure. And so I mentioned that in the beginning of the talk that microtubules are very important for cell division. But what about in a cell? What about in a cell that doesn't divide? Are microtubules still important? Yes, they are. Microtubules are very important for transporting cargo throughout such a polarized structure as a sensory neuron. And so what is then being demonstrated here is that microtubules are used as a track to transport cargo, such as an endosome or an organelle like a mitochondria. So microtubules in this case would be used to transport a mitochondria throughout the cell to deliver energy. And so what these authors did is they wondered, does paclitaxel alter the transport of mitochondria? And so what is being shown here is an image using fluorescence confocal microscopy. You're looking at sensory neurons and that have mitochondria labeled with mito-DS red. And so these are what I'm showing here are chymographs. So the way this works is, so here you see a picture of the sensory neuron axon, and then a picture is taken at certain intervals, and then these pictures are layered, stacked one on top of the other. So going from top to bottom, you see the progression of time. And so when these images are all put together, you can see as you go down this image, you can see streaks going from left to right, and these are mitochondria in motion. And so what the authors measured here was the percent time in motion of the mitochondria. And what they showed is that it was only the retrograde transport toward the cell body that showed a significant decrease or a significant increase in retrograde transport in the paclitaxel treated sensory neurons. And then as far as the speed of transport of mitochondria, there was a significant decrease in the paclitaxel treated in the anterograde, and then the retrograde toward the cell body, there was a significant increase in the speed. When looking at the same paclitaxel concentration, but treated for 3 days instead of 1 day, which I showed at the top, you can see there's no significant difference in the percent time in motion of the mitochondria in anterograde or retrograde movement. And then when looking at the speed, there's no significant difference in the anterograde, whereas there is a significant increase in the speed of retrograde movement. So these findings altogether were pretty inconsistent and they're not very conclusive. That didn't really give any, extreme or significant suggestions that this mitochondria transport could be involved in causing sensory neuron dysfunction. Further, what these authors determined was that paclitaxel's effects are local to the distal endings. So what do I mean by that? So remember that I showed in the diagrams before that it's the terminal nerve arbors innervating the epidermis that degenerate and then progress toward the midline. And so what these authors first looked at was the normal growth of the sensory axons. So in cell culture, the terminal arbors of the sensory axons are having this outgrowth. And what you're seeing over time, it's depicted by the change in color from red all the way to magenta. And you can see this, so it's kind of like a time lapse in one image. And you can see in the control over time, these sensory neuron terminals were growing. And then you see here in the paclitaxel treatment, there's little to no outgrowth at all. So it's really affecting the outgrowth of those distal terminal endings. And so this was quantified at 2.5 hours, 14 hours, and 38 hours of treatment. And you can see in the controls in black that there is growth over time. But however, in the paclitaxel treated dorsal root ganglion neurons, there is little to no growth over time. So there's a significant difference between the control and the paclitaxel treated sensory neurons. And so next, what they wanted to know are paclitaxel's effects specific to a certain portion of the sensory neurons. So the authors had these chambers where they were able to isolate different sections of the sensory neurons. And so they were able to treat certain channels. So they separated these sensory neurons almost like with canals. And what they showed was that when treating the cell body or the mid-axon channel, there was little to no effects on outgrowth. However, when treated at the distal terminal, there was a significant effect on outgrowth, showing that paclitaxel's effects are really local to the distal endings. Okay, so that's the overview of what is known about chemotherapy-induced peripheral neuropathy, and specifically in regard to paclitaxel. Now, and I've talked about some clinical studies and rodent studies, but what about other models of paclitaxel-induced peripheral neuropathy? So there are many non-mammalian models of paclitaxel-induced peripheral neuropathy, or chemotherapy-induced peripheral neuropathy, rather. And there are many advantages to using non-mammalian models, such as Drosophila. And what is common among these models that I'm going to show is they have a high fecundity, meaning they can have a high amount of offspring to allow for really good statistics and lots of observations in a short amount of time. And so in one model of paclitaxel-induced neurotoxicity in Drosophila, it was found they were looking at thermal latency. And so they were using Drosophila larvae. And so what they did was they applied a stimulus temperature, and they showed how the withdrawal of these larvae increased as the temperature went up. So they were very sensitive to this increased temperature. And they showed with paclitaxel treatment that the larvae were much more sensitive to the temperature stimulus, showing that this almost in a way mimics the neuropathy because there's this now increased touch sensitivity. And also during Drosophila larval development, there's also these sensory axon outgrowth. And so what they showed is that the dendritic field in paclitaxel treated larvae was significantly decreased. And these terminal branches also were significantly greater. The number of terminal branches was significantly greater in the paclitaxel treated, showing that paclitaxel is not only, it's interfering with this terminal outgrowth that happens during development. So this presented the idea that Drosophila could be used as a model for paclitaxel neurotoxicity. Another non-mammalian model is C. elegans. Now, this publication was not focused specifically on paclitaxel neurotoxicity, but paclitaxel was used in the study to show that paclitaxel would cause a nerve injury in C. elegans. And so that is what is shown here, that the percent of fragmented axons in the C. elegans was significantly increased in the presence of Taxol. There aren't many studies outside of this that use paclitaxel in C. elegans to study its neurotoxic effects, but this presents a really good starting point and that these models should be used in the future. Now, our lab actually established a zebrafish model of paclitaxel neurotoxicity. And so if you don't know much about zebrafish, zebrafish share about 70% of disease-causing genes in humans. So what I mean by that is if there is a disease-causing gene, there's a really good chance that you can find that in zebrafish and study its function. Zebrafish develop from the yolk sac, so you can view them during development. And you can see here, these are zebrafish larvae that are 2 days post-fertilization. And so you can see they're very optically clear, making them very good for imaging studies. For example, right here, just with bright field microscopy, you can see that their hearts are beating, and you can see that very clearly. But they're also very useful for in vivo confocal fluorescence imaging. And so what you are seeing here is a zebrafish larvae expressing green fluorescent protein in the sensory neurons. And so what we can do here is using the distal tail fin as kind of an analogous part to the human hands and feet. We can see here that paclitaxel over time causes the sensory axon degeneration as shown in humans. So this presented a good model to us to study paclitaxel neurotoxicity. So what our lab found in this publication from 2016 is that paclitaxel causes the upregulation of this matrix degrading enzyme known as MMP13. MMP13 belongs to a family of MMPs, and MMP13 specifically degrades collagen, which is very abundant in the skin. And what our lab found was that paclitaxel-treated zebrafish show an increase in MMP13 expression in the basal skin. And remember, I said it's that the basal epidermis, or it's the epidermis that ensheathes those sensory neuron endings. So what you're seeing here is the basal skin. The keratinocytes are labeled in, the membranes are labeled in green. The nuclei are in blue, and then MMP13 is labeled in red. So you can see there's an upregulation or increased expression of MMP13 in the paclitaxel-treated zebrafish. And interestingly, what we found is that inhibition of MMP13 prevents significant axon degeneration in larval zebrafish. So here you are seeing the axon branch number in vehicle treated, and then compared to the paclitaxel treated fish, which have significant decrease in axon branch number. And then, however, if we co-administer either of these MMP13 inhibitors, we actually prevent paclitaxel-induced sensory axon degeneration. And so, consistent with increased MMP13 expression in our larval zebrafish, we also see skin degradation in the distal tail fin, and also here in the skin, showing us that it not only is MMP13 expressed highly, but we are also seeing evidence of increased MMP13 activity. So given that these free nerve endings are ensheathed by the keratinocytes, and given that we see skin damage prior to axon degeneration, and given that if we prevent MMP13 from its activity of degrading collagen, and then that prevents sensory axon degeneration, this suggested to us that perhaps the skin damage underlies the sensory axon degeneration. Okay, so that is kind of the foundation for what led into my third chapter, and which was part of my main work. And so we'll move on to chapter 3. So the next question was, how does paclitaxel increase MMP13 activity? So MMP13, like the rest of its family of MMPs, its activity is increased by reactive oxygen species, particularly hydrogen peroxide. And so, the main goal of this publication here from our lab was to see how does paclitaxel increase that MMP13 activity. And so we wanted to know, perhaps, was there an increased source of oxidative stress in the skin upregulating MMP13 activity? And so first, what we did is we used a genetically encoded hydrogen peroxide sensor called HYPER, and we expressed it specifically in the basal skin layer of larval zebrafish. And so HYPER can be excited with 2 different lasers. When it's excited with a 488 laser, what you are seeing is the fluorescent signal shown by the oxidized form of the sensor, which has come in contact with hydrogen peroxide. And then the unoxidized form of the sensor, which has not come in contact with hydrogen peroxide, is then excited with a 405 laser. And what can be done is one can compute a ratiometric image to get relative levels of oxidation due to hydrogen peroxide. And so here what you're seeing are heat maps of the distal tail fin of larval zebrafish treated with paclitaxel for 3 hours and 48 hours. And so you can see here in the paclitaxel treated animals in this kind of this heat map that the keratinocytes, or the skin cells, show an increased amount of hydrogen peroxide compared to the vehicle controls. And this is very interesting because we showed in 2016 in our publication, where we established the zebrafish model, that there is a significant increase in MMP13 levels. So this 3-hour time point corresponds to what we also know, there is increased MMP13. And then at 48 hours, we're also seeing that increased hydrogen peroxide as well. And so when we quantify this, we see there's a significant increase in hydrogen peroxide levels in paclitaxel treated animals at 3 hours and 48 hours. So I mentioned, one more slide. So this was at a single time point. So it's important to know whether or not this increased levels of hydrogen peroxide is chronic. So what we did is we did a time-lapse imaging over about 30 minutes, and we looked at an acute treatment. So this is 5 hours of treatment only, and we can see that there's a significant increase in hydrogen peroxide levels consistently over time, just at the acute treatment time point. We see that also at 48 hours. And interestingly, the significantly increased levels of hydrogen peroxide in the paclitaxel treated animals also mimic an injury response. So what you're seeing in blue is the same as on the left. It's the paclitaxel treated. And then in the black, this is the control. But then in red here, what you are seeing is the hydrogen peroxide levels in an amputated tail fin. And you can see how that increases over time. And it's very similar to the paclitaxel treated animals. And also noting that paclitaxel mimics an injury response. So I mentioned before that we had found that when looking at MMP13 protein levels in 2016 in our publication in PNAS, we found a significant level of significant increase in MMP13 in paclitaxel-treated animals. But now we want to know, since we also see a significant increase in hydrogen peroxide at 48 hours, do we also see increased MMP13? And so we did this by doing a Western blot analysis, and we showed that compared to vehicle control, paclitaxel-treated animals have a significantly increased level of MMP13. Now, if we treat with hydrogen peroxide, as we expected, in 2 of our biological replicates, there's a significant increase in MMP13 levels. And then you can see 2 of the biological replicates showed a decrease. And perhaps this was a compensatory effect because of how much hydrogen peroxide was probably being administered to the zebrafish, along with all the hydrogen peroxide that was occurring due to the paclitaxel treatment. We also co-administered N-acetylcysteine with paclitaxel. N-acetylcysteine is a reactive oxygen species scavenger. And so as we hypothesized, if we scavenge reactive oxygen species, we actually can prevent the upregulation or the increased expression of MMP13. And we also did the same with another reactive oxygen species scavenger known as DPI. And so when you administer paclitaxel, and then you would expect increases in hydrogen peroxide and then increased MMP13. But since we scavenged that reactive oxygen species again, we prevented the increase in MMP13. Given that mitochondrial dysfunction was also implicated, we tried to see if we could simulate mitochondrial ROS increase and if that would affect MMP13. And here you're seeing paclitaxel plus rotenone. And then we did rotenone alone. Now, this was the maximum tolerated dose of rotenone, but we did not see an increase in MMP13. This could be due to the maybe it did not affect the mitochondria as strong as we expected. But here, using another mitochondrial ROS inducer known as paraquat, we actually did see a significant increase compared to the control of MMP13 expression. And paraquat alone also induced significant increase in the expression of MMP13. So overall, what this showed us is that in the zebrafish, MMP13 can be regulated by reactive oxygen species, and then also can be regulated by reactive oxygen species produced by mitochondria. So given that I showed in the beginning that MMP13 activity is increased, we see consistent with that there is skin damage, but we wanted to look a little closer at the skin to see what kind of damage it was causing. And so to do that, we used transmission electron microscopy. And so what you are seeing here in the zebrafish skin are the different layers of the skin. And here in the yellow, arrows are pointing to the basement membrane of the epidermis. And you can see that this is a nice bold line here of the basement membrane. And then here in the paclitaxel treated animals, you can see that basement membrane, the density is much less. This is the basal layer of the epidermis in green, and then P is the periderm. And then ensheathed by those 2 layers, you can see in the blue and the yellow, these are sensory axons. And then here is another image showing that a lack of density in the basement membrane layer. And then interestingly, when we co-administer MMP13 inhibitors, we also see a recovery of the density of that basement membrane. So we can see not only is paclitaxel causing this superficial damage of the epidermis, but also at the interbasal levels as well. So given that reactive oxygen species can regulate MMP13 activity, and we know sensory axon degeneration is MMP13 dependent, we want to know, could ROS or reactive oxygen species also regulate the axon degeneration itself? So what we did is we used in vivo confocal microscopy, and we treated zebrafish, larval zebrafish with paclitaxel for 96 hours, as we previously showed in our 2016 publication. That is a time point when there is significant axon degeneration. And so we treated with various compounds, paclitaxel plus NAC, the ROS scavenger, and then rotenone and paraquat, which would induce mitochondrial ROS. And so when we quantify the absolute branch length, as expected, the paclitaxel compared to the vehicle shows a significant decrease in absolute branch length. When we co-administer N-acetylcysteine to scavenge ROS, we still see decreased absolute branch length, but it's not as low as paclitaxel alone. So we're able to rescue some of that degeneration by scavenging the ROS. And then with the rhodonor paraquat, which are going to induce mitochondrial ROS, we see a significant decrease in the absolute branch length. So overall, this indicated that reactive oxygen species alone, if we just increase that on its own, it can go on to induce paclitaxel-induced axon degeneration. And this is consistent what's been shown in rodent models, that oxidative stress is really mitigating this entire phenomenon. So, and now we wanted to look closer at mitochondria. Can we see actually morphologies of mitochondria? If we look closer at the morphology of mitochondria, can we see signs of dysfunction that maybe mitochondria are a source of reactive oxygen species leading to this axon degeneration? So again, we used in vivo confocal microscopy, and we looked at mitochondria in the skin. And so you can see here in the vehicle versus the paclitaxel. In the paclitaxel treated animals, the mitochondria are magenta, and then in blue, that's the keratinocyte nuclei. We can see that the mitochondria appear a little more elongated and a little thicker compared to the vehicle, where they're a little more punctate and not as long. And then if we look at, and so this is an acute treatment at 3 hours, and then we looked at 96 hours when there's significant axon degeneration. You see the mitochondria are pretty punctate, a little more round. And then in the paclitaxel treated, they are a little more elongated, a little more filamentous. And so if we quantify this length width ratio, we can see that the paclitaxel treated animals have a significant increase in this length width ratio. So they are less round, they're more long. And so this is consistent what's been shown in rodents and in vitro cell culture, that the mitochondria in the presence of paclitaxel have this swollen morphology, indicating some sort of dysfunction. So we wanted to look closer at the mitochondria using transmission electron microscopy. And so what we saw is that when we looked at mitochondria in the skin, in vehicle versus paclitaxel treated animals, we can see there's a much more dense outer membrane, and you can see these inner folds that are expected to be seen in mitochondria. Mitochondria have an inner and outer membrane. And then you can see in the paclitaxel treated animals that this inner membrane is much more diffuse, indicating that there is some sort of dysfunction and damage to the mitochondria. We didn't want to be biased though and just look in the skin. So we decided to also look in the sensory axons. So you're looking at the sensory axons of the larval zebrafish in green, and then in red, you are seeing the mitochondria. And when we quantified the morphology, like we did in the skin, we saw an increased diameter just at these acute time points. We see increased mitochondrial length. That is significant, and a significantly increased length width ratio. And there was also an increase in the number of mitochondria per branch segment of the sensory axons. And then as the branch length, as we go along the branch length, as that increased, we also see increased numbers of mitochondria. And to us, this suggested perhaps mitochondria are compensating for the damage that is occurring in the sensory axons. And so like in the skin, we wanted to also look at the hydrogen peroxide levels in the sensory axons. But actually first, I got ahead of myself. We also want to look closer at that mitochondrial morphology using transmission electron microscopy. So when we look at the mitochondria in the sensory axons, we see in the vehicle, we looked at 3 hours at acute treatment and 96 hours when there's significant axon degeneration. And you can see the mitochondria here. You can see those inner folds. And if we look at paclitaxel treated animals, we see that the mitochondria in the sensory axons, if you look at these arrows, they show these signs of vacuoles. And so I showed way in the beginning in rodents that this also occurred. There was this vacuolation of sensory axon mitochondria. So we're seeing in the zebrafish also what has been seen in rodents. Now, interestingly, when we treated with MMP13 inhibitor, and remember when we treat with MMP13 inhibitor, we prevent significant axon degeneration due to paclitaxel. We can actually, we prevent that. But at 3 hours and even 96 hours, when that significant axon degeneration is occurring, we still see signs of vacuolation of the mitochondria. And so what this says to us is that the vacuolation of the mitochondria or the damage itself is not causal to axon degeneration. And so, like I said, we wanted to also look at the sensory axon hydrogen peroxide. And so what we found using the hyperimaging and the sensory axons is that when we quantify the hyper ratio, is at 3 hours, there's actually a significant decrease in the hydrogen peroxide in the paclitaxel treated animals. And then there is no significant difference at 48 hours and at 96 hours. And to us, this is suggested that perhaps that if there is increased hydrogen peroxide or oxidative stress in the sensory axons, that the sensory axons are able to compensate for this. And perhaps what we're seeing even at 3 hours is an over compensation for this increased oxidative stress. Perhaps there's this upregulation of antioxidant enzymes to ensure that the sensory axons aren't experiencing such oxidative stress. So we know that MMP13 inhibition prevented sensory axon degeneration in our zebrafish model, but in rodents, does this also prevent neuropathy symptoms? We want to see if the MMP13's activity was conserved. So to do this, we used a von Frey assay. And a von Frey assay is a behavioral assay used in rodents where there is a filament that is used to assess the touch sensitivity of the paws. And these filaments can differ in thickness. So one can progressively increase the thickness or increase or decrease the thickness of these filaments to assess touch sensitivity. So first we assess this in rats. And what you are seeing here, is actually thermal latency. So we use the heat stimulus. Later on, I'll show the touch sensitivity, but it's the same sort of concept. And so here in black is the vehicle, is the vehicle plus the inhibitor, and then in purple is the just vehicle alone. And then you have the paclitaxel plus varying levels of the inhibitor, and then paclitaxel alone in red. And if you just focus on the vehicles in purple and black, you can see compared to the red is significantly lower. And this is indicating that there is a much more sensitive reaction to the thermal stimulus in the paclitaxel treated animals. However, when we co-administered paclitaxel and MMP13 inhibitor, you can see that there is a recovery of this thermal sensitivity. And then we did the same with cold latency. There's a similar pattern where there's increased sensitivity to the cold in the paclitaxel treated animals. And then when co-administering paclitaxel in the MMP13 inhibitor, you see a recovery of those symptoms toward the vehicle controls. And then this last one is what I'd actually described in the beginning. It's that tactile threshold. And you can see where the paclitaxel treated animals have a very low threshold, meaning they're very sensitive to touch, whereas compared to the vehicle and inhibitor, you can see the touch threshold is much higher, and the co-administration of paclitaxel and the inhibitor brings that touch sensitivity back up toward the control. And so it is known that paclitaxel can have different effects in rats and mice, so we decided to also look at this in mice. And so we can see here a similar pattern. This time the paclitaxel is in blue. And so you can see here both of the vehicles are in black and gray. And that co-administration of paclitaxel plus the inhibitor brings us from the blue to the red, and we start to see a recovery of that increased sensitivity. So we wanted to know where exactly was MMP13 acting? What could it be coming from the sensory neurons themselves? So we wanted to kind of parse out the skin and the sensory neurons. So in collaboration with Francesco Lotti at Columbia University, we co-administered paclitaxel and MMP13 inhibitors to dorsal root ganglion neurons in cell culture. And so what Francesco did here is looking at the sensory neurons in cell culture. When we administer paclitaxel compared to the control, you see there's a decrease in that neurite length. But when we administer paclitaxel with either of these MMP13 inhibitors, and these are the same inhibitors that prevented sensory axon degeneration in our zebrafish in the beginning, and we quantify this, what we show is that just looking at the control here, the paclitaxel has a significant decreased neurite length, but when we have kind of this increase in dose of the MMP13 inhibitor, we see there is no effect on the neurite length, meaning that the paclitaxel is still causing significantly decreased neurite length. And it's the same for the other inhibitor as well. So what this is showing us is that this MMP13 activity that we can prevent, that prevents neuropathy symptoms, and that when we inhibit its activity, it prevents sensory axon degeneration in the zebrafish. When we look at the effects in cell culture alone, the MMP13 inhibitor has no effect. And what this suggested to us that the activity of MMP13 is axon extrinsic. So in summary from these findings, what we can conclude is that paclitaxel is damaging the epidermal keratinocytes. It's causing a breakdown in the membrane barrier. And then we see dysfunction in the mitochondria. We see signs of dysfunction indicated by the swelling morphology. And we also see increased reactive oxygen species in the epidermis. And we think either of these and or the mitochondria, or, well, we know the hydrogen peroxide also maybe combined are causing an increased MMP13 activity. And then this is what is leading to sensory axon degeneration. And then we know from what we saw in sensory axons that even though there's signs of mitochondrial damage indicated by vacuolation, that there is no increase in the hydrogen peroxide in the sensory axons. So in summary, mitochondrial damage does not appear to have a primary role in sensory axons, at least. Whether or not the mitochondria are the source of reactive oxygen species in the skin, or it's another source causing the increase in reactive oxygen species, or particularly hydrogen peroxide, is still yet to be determined. And MMP13 appears to be neuron extrinsic and conserved from zebrafish to rodents. Okay, so that was my third chapter, and now I'm going to talk about my fourth chapter, longitudinal RNA sequencing of skin and dorsal root ganglion neurons in mice with paclitaxel-induced peripheral neuropathy. So as I just went over in my third chapter, we showed that in rats and mice that co-administration of paclitaxel and MMP13 inhibitors prevents these neuropathy symptoms. And so what we wanted to do is further examine what is happening at the transcriptional level in skin and sensory neurons, and what can we learn about what is occurring during this course of development of neuropathy in rodents by looking closer at the transcript levels in either of these tissues. So in collaboration with Dr. Benjamin Harrison from the University of New England, we currently have a study under review where we performed longitudinal RNA sequencing of skin and neurons over the course of development of this condition. So we had mice that were treated with vehicle and paclitaxel. And so we looked at 4 distinct time points. Day 4, which is the pain onset. Day 7 is the pain maximum. And then we have the pain resolution at day 11, and then the recovery of pain at day 23. And so at each of these time points, mice that were treated with paclitaxel, we had skin and dorsal root ganglion neurons that were isolated. And then over at the University of New England, the libraries were prepared, sequencing was performed, and all of the sequences were aligned. And then I myself took the raw gene counts and performed the analysis using R and the package DE-seq2. And this was just a behavioral study that you've seen before, this withdrawal threshold. And it was just to make sure that we were actually inducing neuropathy in the mice in which we were collecting samples from. And then, so this is just an example of preliminary analysis, but we can see this is a list of significantly regulated genes at the various time points and the skin and dorsal root ganglion neurons. And so from here, we are now performing a subsequent analysis for another publication where we actually treated these mice with MMP13 inhibitors. So we can actually get an even more in-depth look at what is happening upstream and downstream of MMP13 activity over the course of development of neuropathy. So that will be coming in the follow-up publication after this. And I should mention this is a public data set, so anyone can access this data set now. And so we believe this will be a really good resource to the peripheral neuropathy community, especially those studying toxic neuropathy. And so, and just to do some kind of quality control, what we see in this data is what we hope to see. What we expected was that, so you can see on the right in the skin, we look at a housekeeping gene and then a validation gene. And we see that at that time point of peak pain, there's a significant increase in the transcripts for MMP13. And then as we would expect in the dorsal root ganglion neurons, there's actually no significant increase in the levels of MMP13. And this also goes with what has been shown in the literature is that MMPs are known to be expressed at very, very low levels. And so their expression is very tightly controlled. And you can see the expression levels are quite low. Okay, and so now I will conclude. So in conclusion, what I showed was that our evidence from our lab suggests that paclitaxel is damaging the epidermal keratinocytes, and it is this damage to the epidermal keratinocytes that are ensheathing these sensory axons that goes on to cause the sensory axon damage. And this is caused by we see increased reactive oxygen species in the skin. We see signs of mitochondrial damage, and we believe this is happening upstream of MMP13 activity. But of course, the exact source of reactive oxygen species or hydrogen peroxide has yet to be determined, and that will be focused on in the future. So what could be some other sources of hydrogen peroxide? Well, like I had mentioned before, perhaps the mitochondria. Mitochondria are exhibiting some morphology changes indicative of damage, and so some future studies will look more at the mitochondria as a source of hydrogen peroxide. Perhaps immune cells. It has been shown that paclitaxel upregulates cytokines or factors that are responsible for recruiting immune cells, and immune cells are known to have an oxidative burst during the immune response. So one suggestion is that perhaps immune cells could be contributing to the oxidative stress, but it is also known that in patients that receive paclitaxel that they experience neutropenia or a lack of white blood cells. So perhaps even if it is found that immune cells can contribute ROS to the skin, that perhaps it is only a minimal effect, but that has yet to be determined. Another source of reactive oxygen species that will be looked at in the future are NADPH oxidases. These are membrane-bound enzymes found in all epithelial tissues, including the skin, that are constantly producing reactive oxygen species. And so this is another source of reactive oxygen species that potentially can be looked at as a source of increased oxidative stress in the skin in the future in the presence of paclitaxel. And then neural epithelial interactions. So I showed previously in our collaboration with Francesco Lotti that the MMP13 activity appears to be axon extrinsic. There are many studies, actually recent studies in Drosophila, that have shown the importance of the neuron extrinsic environment in the development of sensory axon degeneration in the presence of paclitaxel. So we believe that our study will help contribute to the knowledge of the interactions between the outer epithelium, the keratinocytes surrounding the sensory neurons. And there are many tools out there that can be used with our data set that is now public. One is Cell Phone DB, and this will allow someone to take our data set and look at the ligand receptor interactions occurring from tissue to tissue based on the expression of certain genes in the tissue that they stem from, and also NicheNet, which can do pretty much the same thing. And these are both stemming from different public repositories of ligand receptor interactions. And possibly through this analysis, there can be some new key players revealed in this whole entire process. So with that, I would like to thank our collaborators, our former lab members, of course, Ricardo Cepeda for keeping all of our fish alive and really helping us maintain the animals that really bring our research forward. The Rieger Lab, my thesis committee, and all of my fellow biograds, thank you for all your support. And of course, my thesis advisor, Dr. Sandra Rieger. She mentioned at the beginning all the opportunity I've had over the past 5 to 6 years. And without her, I wouldn't be here. So thank you very much. And I'll take any questions. Thank you.
Thank you very much for this excellent talk. Are there any questions? I guess if there are no more questions, then we can just adjourn to our private meeting.
Thank you very much, everybody.
Okay, thank you.
Nice job, Anthony.
Thank you. Thank you for coming. Okay.
Congrats, man.
Thank you very much.
Congratulations.